All right, I think it's about the time to start. If you are ready, Ellen.
Yes, I am.
Very good. So good afternoon or good morning. I'm Alfio Quarteroni, the MOX director, and I'm very pleased to have today for our MOX Colloquium Professor Ellen Kuhl from Stanford. Ellen is the Robert Bosch Chair of Mechanical Engineering at Stanford University, and she is from Germany, from the northern part of Germany. She received her PhD from the University of Stuttgart in 2000, and her habilitation from the University of Kaiserslautern in Germany in 2004. Ellen works primarily, I would say, in the design of models, both the theoretical computational design of models to simulate the behavior of living structures. She published more than 200 peer-reviewed journal articles. She's a fellow of the American Society of Mechanical Engineers and the American Institute for Mechanical and Biological Engineering. And she received the Humboldt Research Award in 2016. This is a very prestigious award in Germany. She is a founding member of the Living Heart Project, which is a translational realistic simulation with several hundreds of participants from different countries and different environments, research, environment, the industry, and medicine. Well, Ellen is also a multiple marathon runner of Boston, Chicago, and New York Marathon, and a Kona Ironman World Championship finisher. And you understand that with these premises, you'll not be surprised to know that Ellen has immediately accepted our invitation to speak in our colloquium, even though it is 5 a.m. in Stanford. So very remarkable. The title of her talk today is Modern Dementia. So it's a very important subject that I guess has to see with neurodegeneration of brain. And so Ellen, thank you very much for accepting to give the MOX Colloquium today, and now the virtual floor is yours.
Thank you, Alfio, and thank you for inviting me. Let me share my screen. So if you can see this, so I would like to talk about modeling dementia, and what you can see here, we were just joking, it's not a test screen. I'm going to tell you in just a bit what it is. Think about what you would think it is, and then I'll try to answer your question towards the end of the talk. So this is actually the work of a PhD student here at Stanford, Amelie Schäfer, and it has been supported by a lot of other people. So Sveva Fornari is actually a student from EPFL who did her master thesis with us. Taylor Harris is a student from Stanford. Louise Noel is a Belgian postdoc. Johannes Weikemeyer is a postdoc from ETH, who is now a professor at Stevens Institute of Technology in New Jersey. And then Matthias Jucker is a real expert on dementia. So he leads the Dementia Center in Germany for the entire country, and he's situated in Tübingen. And then Alain Gurrieli, many of you probably know, he's a professor of mathematics at Oxford. So I would like to talk about Alzheimer's disease, and many of us know a lot about it, but just to introduce you a little bit to the subject. So you can see here on the top, Alois Alzheimer and his first patient, Auguste Deter. And in between them, you can see when Auguste Deter visited him for the first time, this is her handwriting. And this is a very typical handwriting of Alzheimer's patients. And it's very crumbly and very repetitive. And so Alzheimer noticed immediately that something was not right with her. She was in her early 40s. And so he said, okay, I would like to examine her brain if she passes away. So that took a couple of years. And she always claimed that she had forgetfulness. And so that was actually the disease that characterized it the most. And she passed away in 1906. And that's when Alois Alzheimer got her brain and did a very thoughtful pathology. So he cut the brain, stained it, and he found what you can see on the bottom of this picture. So this is really from her brain. So on the bottom left, you see amyloid beta, and these are proteins that clump up in the brain. And on the right, you see what's called tau tangles. And actually on the top right, you see a drawing that Alzheimer made of these tau tangles. And interestingly, these two things are still to this very day the two hallmarks of Alzheimer's disease and how to diagnose it. And the most really conclusive diagnosis is by cutting the brain, staining, and looking at these two proteins, even more than 100 years later. So this is still remained very important. What is added to this? So back then, she was the first patient to find. Now, every 3 seconds, a person is diagnosed with dementia in the world. So this is a real serious problem. The number of people with Alzheimer's disease will triple by the middle of this century. And this is just a huge financial burden. And if you think about this, there'll be 130 million people diagnosed. So many people around us by the middle of the century will actually have Alzheimer's disease, maybe even us, so we have to care for a couple of people. So this is really, really severe. The problem with it is that we can't diagnose it timely. And this is from a medical paper. And this is how people imagine that actually Alzheimer happens. And this is just a drawing. So it's not a model or anything. It's just how people imagine that this happens. So in red is this curve that shows you how amyloid beta evolves. And in gray underneath is the detection threshold. So it evolves very, very slowly. And once it starts to peak up, you can detect it once it passes a threshold. With a little time delay, so on the y-axis, x-axis, you see the time, come the tau proteins that we've actually seen. So that's the two clumping proteins. And also first you can't detect them, and then all of a sudden they ramp up. We can only actually first notice it with this orange line, and that's brain atrophy. So that's a shrinking of the brain. And then with a little time delay again, we see what actually we can diagnose as in the clinic, that's cognitive impairment. So when people come and say, really, I tend to forget things. So when Augusta Data came to see Alzheimer's, so she actually was in this green curve, already in this very green region. And many of her brain has already either shrunk or had a very severe content of amyloid beta or tau. So it's a real problem that the diagnosis lags by 15 to 20 years. And to date, there's no way of diagnosing it through imaging. So you have to cut the brain and slice it, which of course nobody wants to do on a living brain. So remember this curve, I'll come back to that later in the talk. There was a breakthrough in understanding Alzheimer's disease about 8 years ago. And that was actually led by the person that I mentioned at the beginning, Matthias Jucker. And they recognized, or in this nice review article, summarized that human neurodegenerative diseases are prion-like. So many of us would probably not know what prions are, and it's shown on the top right. So prions are proteins that actually misfold, and that's a disease mechanism that was discovered in 1997 by Stanley Prusiner and received the Nobel Prize. And the idea is that you have a healthy protein and a disease protein, and the disease protein recruits the healthy protein, and then they all misfold on top of one another. This is something that's known to happen in BSE and cattle, in scorpion sheep, or in Creutzfeldt-Jakob disease. And this is actually always very, very slow. It's not treatable, it's progressive and always fatal. So these four things are characteristic. Now, human neurodegenerative diseases like Alzheimer's, Parkinson's, or ALS are not exactly prion diseases, but they are prion-like. So the mechanisms that underlie these diseases are believed to be similar to this. And so this is actually what we want to model. Here you can see the picture of amyloid and tau on the top that you've already met in the second slide. And underneath you can see alpha-synuclein. This is in Parkinson's disease, and TDP43. And this is in ALS. So you can see similar features of these clotted proteins. So now what was summarized in this nice paper by Matthias Jucker and his colleague here in the United States, Walker, is the staging of these brains. So these are time points of different diseases, and they start at a different part of the brain, and from there they spread across the entire brain. So you can see, for instance, tau, the blue one, starts somewhere in the brain stem where it's blue initially. Then it goes to the front of the brain, and then it spreads over the top of the brain to the back. So the part that's affected the last is really in the back of the brain. And you can see this here for all other diseases as well. There's a very characteristic stereotypical spreading pattern that is very repetitive across these diseases. So now when we look at this from a modeling point of view, there's two things. There's once this accumulation of clots, which is a local phenomenon. And then there's this spreading through the brain, which is global. And so now we're modeling people. So the local thing we model through ordinary differential equations that describe how much protein is clotted. And then the spreading we describe through a partial differential equation, just simply by diffusion through the brain. I'll start with a local equation and actually make it very simple because there's very little known about the kinetics in the real brain. A lot of people do experiments in a petri dish, but those don't necessarily translate to the brain. So what you can see here is a very simplest assumption. So you have in blue a healthy protein and underneath in red a misfolded. So for the healthy, I use the letter P, and for misfolded, just a P with a little tilde for the misfolding symbol on top. So the healthy protein tacks onto the misfolded protein. They both misfold. So we have two misfolded proteins, and then they actually disassemble again. So now we have two. If you were to do this with real reaction kinetics, you need kinetics for recruitment, misfolding, and fragmentation, these three steps. And that's somewhat too complicated and also not necessary to do. So what we do is we just describe from the healthy to the disease state, everything in ones, a conversion from blue to red. And that makes it a lot simpler. So now this shows what exactly happens around this. So there's a couple of things that happen. And we describe this with these two equations that you see underneath, where the blue equation, or the equation for P, describes the healthy protein, and the red, or with a little tilde, describes the misfolded protein. So there's a production of only healthy protein, so you can't produce it misfolded. And then the healthy protein in a normal healthy brain gets cleared in a regular way, cleared away from the brain. And that's this little K1 that you can see. Then there's also a conversion, and that's what we just talked about, from healthy to diseased by recruiting through this red misfolded protein. And you can also clear either the healthy or the misfolded protein. So this leaves actually 4 constants, and that's important because we want to somehow describe this with real kinetics. So there's a diffusion constant through the brain, and there's 4 kinetic constants: the production of healthy protein, the clearance of healthy and misfolded protein, and the turnover from healthy to misfolded. And you can understand the turnover is obviously the critical part of this. Now, we don't want to describe these 2 equations because it makes it somewhat cumbersome to describe, and we don't actually need to. We can make this actually a little simpler. And what we do is we assume that in the healthy brain, we only have healthy protein when we start this process. So we develop a Taylor series around that starting point, and then we can actually replace the healthy protein concentration from the first equation in the second. So we only have one equation with one unknown, the misfolded protein concentration, P. So that's what you see at the bottom. And then you can jiggle this equation a little bit and make it a little nicer and reformulate it in terms of C, which is a concentration that's scaled from 0 to 1. So all I've done is I've used these 2 equations, eliminated one, and look only at the misfolded concentration. And that leaves this equation that you see in on the bottom. And that's an equation for the concentration that runs from 0 for nothing to 1 to fully misfolded. And when you look at this very carefully, this is a very simple equation because it only has one spreading term, like we wanted to do initially in our motivation, and one growth term. And if you look at the math of this equation is known as the fisher-kolmogorov equation. Now, here's a slide that actually shows the initial paper of Sir Ronald Fisher, and this is well known to mathematics or biomathematics. And this is an equation that actually initially was developed to describe mutations in genetic population. And you can see how this equation looks like. And it's a very, very famous paper and very well-known equation that's been used a lot to look how mutations of whatever spread in a genetic or in any kind of population. So this is a known equation that we're working with here. For us, we can look at its parameters. So one of the parameters obviously is this growth, the alpha, that scales how fast actually we produce misfolded protein. And then we have the diffusion. And I guess I'm talking to a group that knows a lot about diffusion. So I don't need to explain in detail. We have a second order diffusion tensor D. And that is parameterized in 2 terms: an external diffusion, which is an extracellular, so outside the cells, and that's isotropic. It's just an isotropic diffusion. And then we have diffusion along the axons. It's believed that these proteins, some of them live in the axons, so the long cables in the brain, and that they diffuse along the cables. And I will show you in a bit why that actually makes sense. So we have these parameters, and then we have the fiber direction, or the axon direction in the brain. And that's something that's not a free variable we can actually measure on the brain with magnetic resonance. So we discretize this in time. We use finite differences here, and then in space with finite elements. And that brings us to a brain model that you can see here. So this is the brain. Actually, it's my brain, with white as the white matter of the brain. So that's where the axons live. And then around it in brown, you have the gray matter. And we have a couple of different models. So we have a whole brain model. That's a three-dimensional brain model that was recreated from magnetic resonance images. We have a sagittal model. So that's a slice crossing the brain from left to right, and a coronal model that's cutting the brain from front to back. So now here's a sensitivity study with just these parameters that we discussed. So the first one is looking at where we seed the misfolded protein. So you can put the initial seed of misfolded protein anywhere in the brain, and then from there you can see how it evolves. So you can see you can produce different patterns of these spreading patterns if you put the initial seed in different locations. So actually that seems to work well, and that seems to do what we want it to do. We can also play a little bit with the parameters. So the first one is a kind of baseline, the first shot at it. And then the second picture and the one on the bottom left increases the diffusion. And you can see that then the proteins just diffuse faster. You have a more homogeneous picture. You don't have dark red to blue, but everything is in the order of bluish and yellow. And then the last one is producing more. So you can see where you initially start, where I seeded the protein. You have a very high concentration and it doesn't spread that fast. So actually you have these parameters to play with. I'll show you at the end of the talk how we actually really identify these parameters for real brain. And this is the last one, and this is what I just said. This is something we don't need to guess. This is known. So this is the fiber orientation. This is just showing how sensitive our simulation would be. So the first picture shows an isotropic diffusion. That if the cells were just the protein were just diffusing isotropically across the brain. The second one has a fiber orientation, a pronounced spreading around the ventricle in this ellipse format. The third is a radial, like a star. And then the fourth is actually what it looks like in the brain. In the real brain, you have fibers that actually circle around the ventricle and then point outwards. So this is more what it looks like in a real brain. So now let's see and test the model. We do this for the first case of tau inclusions in Alzheimer's. And we've already seen this starts in the brain stem and then goes to the front and then propagates to the back. So we seed in the brain stem and we do the simulation on the bottom, and you can see from there, it goes to the front and then it goes to the top. It doesn't reach all the top, which is in agreement with the picture that the clinicians actually provided. And then it goes to the back, which gets activated or misfolded at the last. For the 4 different regions, you can seed in different locations. So we just take these images that I've showed at the very beginning, and we seed where these are in dark color. So we seed in dark orange, blue, red, and green. And then from there, we run the simulation. You can see that you can produce different concentration patterns depending on where you first put the misfolded protein in the brain. So this actually seems to work quite well. If we compare it to the picture that clinical people have provided, then you can see it actually provides a similar pattern and distribution of misfolded proteins just by using this mechanism. So this is nice because we can now in a very simple way describe how the pathology of neurodegeneration spreads across the brain. We just use a reaction equation for this growth, which we just have one parameter alpha, and a diffusion equation where we just have one parameter that actually tells us how fast this is spreading. So then we can actually recreate these curves that you've seen at the beginning. And this is interesting because these were curves that people have just drawn by hand and said, okay, it looks something like this. And they were never really able to quantify this. And with this model, we can easily quantify this. So when we show this talk actually to clinical people, they say, oh, wow, how did you do this sigmoid curve? But it's very simple, of course, if you know how to do finite elements. What you do is you just add up the red elements and that gives you the red curve over time, right? So this is projecting over time, and you can see in a window of about 20 years in the simulation parameterization, you hit from 0 to 1. So this is in real agreement with what people have seen, and it's not just drawing this line, but there's really some math behind it And I mean, if you build it like this, it's very simple, very, very easy to understand and also easy to recreate. This is now for the whole brain. This is a little more complex than in 2 dimensions because the fiber directions are a little more tricky. And this is directions that Johannes Weikenmeier has created from real brain scans. And you can see this is the directions of the fibers really in the brain. So now we allow the proteins to propagate along these fiber directions faster than isotropically. And then you can see the 3-dimensional simulations. It looks a little blurry and there's usually not much to gain in this from a 3-dimensional picture. It's a little more complete, but it actually confirms what we've seen before. You can predict the spreading pattern on a finite element mesh if you were to actually use these directions. And you can see again, it matches the picture that was predicted. Again, if you integrate it, you get the same picture. It's a lot more work to do in 3 dimensions. Simply because you use so many elements, but of course you have a better picture of the left and right parts of the brain. So now we're back at this curve, so we can explain the sigmoid shape of this curve that people had before only just assumed. We can also show this distribution for amyloid beta and for tau for these 2 misfolded proteins. Now the question is, can we actually relate this to the shrinking of the brain, which is the next curve, the orange curve? And so we try to actually couple this. And for those of you who know our group, we usually model a lot growing things. And I know there's maybe many of you are also working on growth. So we just use a growth model where we invert the growth and make it negative growth, so shrinking of the brain. This is what it looks like in reality. So if you were to cut Alzheimer's person's brain, on the left you see a healthy brain, and on the right you see really what happens. So there are characteristic features that are very repetitive. So one of them, for example, is that the valleys and ridges of the outer layer of the brain actually widen in Alzheimer's disease. The outer layer, the gray matter, really becomes much, much thinner, which you can also see. The ventricles are the fluid-filled part of the brain. They grow because the overall structure actually shrinks. And then there are certain regions in the brain that shrink disproportionately much. So this is what this picture shows. And we use the growth model to actually do this. And we use multiplicative decomposition, and many of you are familiar with this. So we use a deformation gradient. We have an elastic tensor, and what we usually use as a growth tensor, we introduce as negative growth or atrophy shrinkage. And we use actually the determinant of the shrinkage to describe the volume change. And then the remaining part is the elastic part that determines the stress. So this is just very standard and very simple. And then you can predict how the brain is shrinking. So this is showing if gray and white matter were to shrink at the same time. So it's a general part of atrophy. And you can see these patterns actually pretty much resemble what we've seen. So you have this widening of these gaps between the folds of the brain. You have the enlargement of the ventricles. You have a thinning of the outer layer. And then in the bottom, you can see stresses develop. This is a similar picture where gray and white matter shrink at different rates. And you can, if you look at the stresses, you actually have stress concentrations between the 2 materials, the gray and the white matter. And this is actually where typically you have a deposition of a lot of misfolded proteins initially. So this is believed to actually be maybe even related. So that's where brain gets damaged the first. So this interface between gray and white matter is very important in the brain. And actually, when you look at stresses and you consider them as 2 different materials, it's kind of clear that there's something going on at this jump. So comparing this again to the picture, it fits nicely. It looks pretty much like what we have predicted. And all these classical features actually are visible in this drawing. So now can we correlate the 2? Can we correlate the protein spreading and the shrinkage? So in the middle, you see an Alzheimer's patient, and you see what we've just discussed. We saw these features really on the scan of the brain. And you can see the ventricles getting bigger, these holes in the brain. And then also the cortex, the outer layer, getting thinner, and a lot of empty spaces on the top around. So on the top, you see the simulation of the biomarker. So that's in our case, tau, which we seed, and then it spreads through the slice. And then on the bottom, you see it shrinking. So in a way, you have this amyloid beta. We can simulate tau spreading through the brain. And where you have a high concentration of tau, the brain is shrinking. And then ultimately, you will have impairment of understanding and memory loss. And that's when we diagnose it. So this is now actually nicely coupling these things. This is just how we do the coupling. And this is just a lot of equations to say that we have the protein spreading, the mechanics, we have a scalar equation for the misfolded protein, an equation for the mechanics, and we couple them, and there's two-way coupling. So we have a concentration-dependent shrinkage, and we have deformation-dependent spreading. So these two cross-couple, and this shows actually most nicely if you linearize it and look at the tangent or the coupling terms. So there's a deformation-dependent spreading in the third equation of the tangent, and then a concentration-dependent shrinkage in the lower part of this matrix. So you can see that one affects the other. So now you can say there's a lot of voodoo. How does it actually work? So this is a simulation that Amelie Schäfer did, and on the top row you can see the concentration. So we see it again in the brainstem. It goes to the front of the brain and propagates to the back. And underneath you can see projected on the real brain, so how it's shrinking. And you see there's quite some shrinking going on, starting in the front of the brain and then propagating to the back. And underneath you can see a stress analysis. So if you integrate all of this, we get this orange sigmoid curve that you've seen, but we can actually also calculate how much the brain is shrinking. And you can imagine this is somewhat a painful, large problem to do, because it's a coupled problem of deformation and tau, and we can't really see a lot more. So what we did is we did it on one slice, it cuts this way, and one slice, it cuts this way. So this is two-dimensional simulations. So what you can see is the increase in orange, this sigmoid increase in tau. And then Amelie also calculated how much the brain is shrinking. And that's the red curve that goes down. So in a period of about 15 years, the brain is shrinking by, I would say, well, for say 10 years, the brain is shrinking by 30% in size. So that seems actually pretty large. You should know also that actually the normal brain, a normal aging person, shrinks about 1% a year, which is frustrating because we lose a lot of brain mass over time. So that's a dashed line. So that's a normal age-related shrinkage. So if we take that out of the equation, then the dotted line is actually just the Alzheimer's related shrinkage. So now you can say this is just a simulation. How does it compare to real life? So this is a study on the left with dots of people brain volume loss. And so there's a zero line, and you can see that actually this looks at how the brain volume of these people has changed over time. And these are all Alzheimer's patients. And you can see they follow this red curve on average. And this is the red curve that Amelie used to actually calibrate the model. And there's a certain range. So the left is the real measured data in this paper that's cited below. And then on the right, you can see actually the simulation. So you can see this actually is realistic. You lose this much brain volume over time if you're an Alzheimer's patient. That's really happening. So this is somewhat actually, not scary if you look at it from this angle. So now at this point, we're saying, okay, this is quite expensive simulations. We would like to do this in 3 dimensions, right? I've said we do it in slices in both cuts, but we want to really do it in 3 dimensions. And so we do probably too much. If we use all these parameters and we describe all this, we don't have all this information to put in modeling. In a way, it's always a balance of what do we actually know, what do we want to do with it. And so then can we reduce this any further? So there's a lot known about different regions in the brain. And there's also a lot known about how they're connected. And this is from connectomics. This is when people study signal processing in the brain. So this is typically electrical engineers or people who look at brain wiring. And they actually know how regions of the brain are connected. And this matrix that you can see here is actually called the connectome, or the connectivity of the brain. And you can see blue regions are regions that are not at all connected. And then you can see in red regions that are wired very closely together. And this is something we know from the brain. And actually this connectome that you're looking at right here is the average connectome of 418 adult brains of the Human Connectome Project. People had until this point not used it for spreading Alzheimer's disease, but really for connectivity and for signal processing in the brain. The brain has 4 lobes, so that's 4 different regions that are very closely, tightly connected. And those are actually the red squares that you see on the diagonal. And then it has 2 halves. So on each half, you have a top and lower square of this matrix. So one is the left-hand side of the brain and one is the right-hand side. On each side, you have these 4 red squares. This is within each lobe of the brain. And then on the left, you can see really how these regions are connected. So there are 83 of them. And you can see the regions that have a high degree of connectivity And then you can actually map that out into this matrix. So this is nice because we can use this for the spreading. So what we now do is we solve these equations instead of solving them on a three-dimensional finite element mesh. We solve them just in this network. The process is just the same. And you can think of this, if you've never worked with networks, as just finite elements. So it's like a truss element gives you the same equation. If you were to discretize this mesh on the left-hand side with truss elements and connect the nodes, then you have trusses in 3D. And the proteins travel along these trusses. Now they're weighted trusses because there's somewhere it travels faster, but this is essentially what this is. And then you can simulate by seeding, again in the brainstem, you can simulate where you have these bubbles pop up, and this is high concentrations in these regions. And actually it matches the picture quite well if you look at where these points lie in the brain. What's nice is we can do a lot of games with this because it's very cheap simulation. So this runs now for 10 seconds or so on any random laptop. And we can integrate these curves very easily, and we can actually integrate them for different brain regions. So for instance, what this shows us, and that's an interesting question to ask, can we reproduce the activation patterns? So in this case, we can see the green part of the brain, that's called the temporal lobe that sits at the bottom, gets activated first, followed by the front, which is the red, the frontal lobe. In orange, the parietal lobe is the top, and then the occipital lobe in blue is the last. And this is actually exactly what we had predicted with the model, in which matches also what you can see. So this is kind of like reconfirming, and it's nice. If you integrate the whole thing, you can see the network model in the gray and the continuum model in black, so they predict the same thing. So if you're only interested in like more global understanding, this is a nice way to do it. And it's very cheap, very simple, and you can play a lot with the parameters. You can, for instance, say which region is the most connected. So this is seeding at all different 83 regions and looking how it spreads across the brain. So there are regions that are very well connected in dark red. From there, it spreads very fast. And then there are regions that are pretty much not connected, the front and the brainstem, from where it spreads very slow. So now that's interesting because it actually starts in the brainstem, in the region that's the least well connected. And that's why it takes so long. And that's this dark blue curve that you can see here, spreading over 25 years. Another thing you can then do, and that's something I haven't told you yet, is actually, this is a very, very simple model where you only look at one misfolded and one healthy protein. In all reality, when we looked at the very images, it's a cluster of misfolded proteins. And so if you're a biochemist, you would actually model the individual clustering. And this you can do with what's called the smoluchowski model, and it's well known actually for biochemistry of these misfolded proteins. So you start with a protein of length 1, and you attach more and more and more misfolded proteins. And so then in the end, you have proteins of certain lengths, and that's what forms a cluster. So now when you model this, of course, you need to keep track of all different lengths and store how many proteins of each length you have in your model at any location. And this is why we use this network model, because a finite element model would just be very expensive. These chains can be up to, say, 200 proteins in length. So at every point, you need to store the 200 lengths, how many of them you have, and then the turnover you calculate. So 200 is just at every location. If you have 83 nodes in the brain or regions, it's 83 times 200. That's easily doable. But if you have, say, 1 million nodes in your three-dimensional finite element mesh, that would be 80 million degrees of freedom all of a sudden. So that makes it very expensive. So this runs well on this brain, and you can see, you can predict these curves now for different sizes of protein length. And this is nice because if you talk to people who treat Alzheimer's, they say, hey, the small molecules, the very short, fragmented things, we can actually probably clear very well with drugs. So there's a lot of drugs that target small molecules, which are the red curves here. And then over time, these red curves disappear, and you actually have these longer proteins. So that's the dark blue forming, and that happens much later. And that's the real clusters that we can see. So we can run this, and we could actually potentially say, what happens if we clear the smaller sizes? What happens if we clear the larger sizes? And this is what the simulation predicts. And all this runs within seconds because we use this very fast network model. Then we can plot it on the brain. And this is nice because we now work with these 83 regions, and they are known to sit in certain places of the brain. And then we can map that back onto the brain surfaces. And you can see, for example, how it would spread across the brain. The first row is just the 2 length proteins. The second row is anything that's larger than 8 in length, and the last is anything that's larger than 16. So you can see it progresses from a short length over time to much longer chains of proteins that are misfolded, so really these clusters or clumps or aggregates. So this is some good understanding actually of what really happens with the kinetics. So now you might say, oh, you're just making all of this up. Can you actually back this up with any data at all? And this is something we just did very recently. So Amelie Schäfer did this, and we just published a month ago. And this was not possible when we started the study because there was no way to actually image tau protein in the brain. And this came up about 3-4 years ago, that people were really confidently saying, we can image misfolded proteins. We're actually working with someone in radiology who does these scans, and we also draw data from a public database. This was the first time when we started the study that there was actually a longitudinal mapping of PET scans over several years. So you can tell this method is just within the last decade, has been developed only in the last couple of years. So what you see on the left is a PET scan. So that's positive electron tomography. So it's a kind of MRI scan where misfolded tau protein lights up. In the middle is a regular MRI scan. So we register these regions on an MRI scan. So we kind of match them up. And then we segment everything. And there we use a software that's called FreeSurfer. That's a freely available software that maps the MRI scan into these 81 different regions. And then in every region, we can read out the tau PET concentration. So this is a pipeline of processing that Amelie now does very well. And she has done that on 46 brains. And these brains are, 26 of them are amyloid positive. So that are known to people to have Alzheimer's. And they all had multiple scans in an annual rhythm. So these people that we're looking at had at least 3 scans, some had 4. And they add more scans as time progresses. So we'll probably in 2 or 3 years have a very nice longitudinal picture of this. But right now there's only these available. So if you do this, you would have guessed this is the picture that you looked at the beginning. So what you see in all the rows, so just digesting this a little bit, there's a lot of information in this. So on the rows, you have these 81 different regions that actually make up the brain. And as columns, you see the 46 people. And every column has a different width. So some are 4 band width and some are 3. So that's the years of the scan. So the first one is always the first year, and then second, third, or fourth. So you see a progression usually within every band from left to right. We've sorted this so that at the top row, you see the region in the brain that has the highest tau PET concentration. And so that's the most red. And then also on the left, you see the patient that had the most tau PET. So the upper triangle, in a way, gives you the regions that were affected the most and the people that were affected the most. Whereas the dark blue, the lower triangle, gives you the least affected people with the least affected regions. So this has been a lot of work, but now that we actually have those data, we can run a parameter identification. And the nice thing of this very simple model is we only have 2 parameters. The one is the spreading, in this case, kappa, and alpha is the local production. And the first row here gives you the diffusion equation. If you discretize it on the network, that's the second equation. This is what it looks like. This L is in a way the matrix that makes up the diffusion coefficient. It's called the graph Laplacian, and that's what we get from the connectivity. So we only have 2 parameters, essentially, kappa, which is the global spreading, and alpha, which is the local growth of protein. We do a parameter identification. We minimize the error between the simulation and the tau PET concentration. So what you can see on these images is the mean of the tau PET concentration across these 83 regions projected onto a brain. And we have put it into this 0 to 1 interval. This gives us the values of 0.21 for the growth and 0.34 for the spreading. And we've done 2 simulations here, which I haven't talked about much, but we did this study initially to actually find out, does it spread along the axons or does it spread isotropically? So there are 2 different simulations, one with intracellular, one with extracellular spreading. And these are the 2 parameters. On the left, you see the spreading along the axons. On the right, you see the same parameter fit with just an extracellular isotropic spreading. If we take the intracellular spreading, we can project forward what happens. So you can see on the top 2 rows, the raw PET scan. So that's really the clinical scan. Underneath, you see it a little bit cleaned up. So this is projected into the interval from 0 to 1, where our simulation lands. And then in the last two rows, you see the intracellular spreading and the extracellular spreading model. It's interesting actually that the features map pretty well. So from year 0, 1 to 2, you can see actually it predicts exactly what you would expect. The PET scan can be recaptured quite nicely if you compare, for instance, year 2, the regions in color are very similar to what we have done with the model. And then with the model, you can also project forward. You could say, okay, if this is a valid assumption, where would it spread? Where would the regions be affected next? And then, of course, the longer you do this in time, the more you see a difference between the two diffusion models. So after 10 years, you can actually already see quite some differences between the intra and the extracellular spreading. So taking all of this together, here's a summary of kind of what we have learned through this. The first finding, at least for us, is that misfolded proteins in various neurodegenerative disorders spread through the brain. And we can model this very simple universal physics-based laws of nonlinear reaction locally and anisotropic diffusion globally. We've shown that actually physics-based modeling can explain different misfolded proteins in different diseases. So that's amyloid beta and tau in Alzheimer's disease, alpha-synuclein in Parkinson's, and TDP-43 in ALS. We've also seen that we can integrate from these patterns the biomarker curves, and they have this nice sigmoid shape, and that actually matches quite well with the timeline of neurodegeneration. And then you might ask, okay, why is this interesting? It's interesting to project actually a timeline of how these diseases propagate, to estimate in time what happens to a society, what happens if a huge group of people has this disease. For instance, if you were to design a clinical trial, where can you sample in the brain? What are important regions to look at? And then also we've shown that we have made first steps to look at what can we do if we clear certain sizes of aggregates. How would that look? So pharmacological intervention. So interesting. I mean, initially when we started this, people were saying, what can you actually do with physics? Why are you using, right? Why is it so cool that you find a physics model to describe this? So physics, for instance, can explain why it is so slow. And that's something that people have always wondered is, why is it starting and it's so slow to develop and we can't stop it and it's just unstoppable, but it happens and we just see it at the very end. So physics can help explain that. It can also, in a way, help explain why is it unstoppable? Where can we interfere? Where can we intervene? At what time point is it good to intervene? Can we detect it earlier? Can also, and that I've shown you too, tease out the two roles of propagating along the axon. So for us, that's an anisotropic diffusion along a particular direction and isotropic diffusion. So if we want to actually target something inside the cell or outside, you would actually can do that with the model quite well. And then also we can somehow project the timeline on an individual basis. Where are people in this disease? Especially if we can correlate it to shrinkage, because shrinkage we can confidently actually diagnose on a scan. So this brings me to the end of the presentation. These are all the people who have somehow one way or the other worked on tau protein with our group. And I would like to thank you all and I'm happy to take questions.
Alfio, you have to unmute.
You have to unmute me. Okay. Oh, now it's fine. Can you hear me now?
Yes.
All right. So for some reason I was muted from the organizer. So thank you very much. This is it's a great talk. So I'd like to ask the participants who want to ask questions to raise their hands. And to start with, I will maybe ask one question. Or of course, there are many questions that could be raised, but I'm wondering from one side, how do you actually reconstruct the direction of the fibers from one hand? And from another hand, if in your one-dimensional model for diffusion, your two basic coefficients, kappa and alpha, if you keep them fixed in time or if they change in time, if they should actually change in time realistically? So this is my first question.
Yeah, that's a good point. So I can just share this one picture again, and that probably gives kind of an idea how difficult it is to do this. So this is the picture of the recreation of these directions. And so the way we have done this, and I think there's now software to do this much better. What we did is we traced the locations of the fibers and then assigned for every element a fiber direction. And this is probably similar to what you do for the heart. So you need to find a point and then you trace it. And then in the end, you register it on the finite element mesh. But this actually gives you kind of an understanding how messy this can become. So it's not all that easy. So we did it for one brain just to kind of see that it works. But you're right, this is a difficult task in a way to do in itself. And I think it's probably similar to when you trace the directions of the heart with a diffusion tensor imaging. Yeah, good point. And then, the time change. Yeah, so these things probably, the parameters probably change in time. So we don't have a whole lot of data. You've seen there's large error bars on these parameters right now. So we're happy to get some kind of sense of how fast are things spreading with real physical understanding. But I think it would be nice to follow these people more in time. So if we have a second, third, fifth year, we can probably more confidently say how things change over time. First of all, there is not such information. And these people only live for about 8 years after it's diagnosed. So the most you can hope for is probably 10 years of resolution, which would already be great. That's a good point, yeah.
I was wondering if they could themselves depend on the solution in some cases, if it's
yeah, it's probably true. They probably do. Yeah, you're right. That's a good point.
Thank you. So Paolo Zunino, you have a question, I guess.
Yes, thank you, Professor Kuhl, for the inspiring talk. My question is about flow. So what about the interaction of your models with any type of flow? I mean, lymphatic flow, cerebrospinal fluid, all these effects, how they could possibly interact with what you presented?
I think that's a great point. So I'm not a biochemist, so I don't really know exactly how this affects these proteins in particular, but I think it's absolutely a valid point to see how things actually, for instance, for clearance, I think flow is probably very important. So the parameters that describe the clearance of these proteins probably would need some, if you do this more realistically, would need some flow. So in general, I think it's a good point. We didn't look at it, but if someone were interested in looking at this, I'm sure you can make the model more realistic by including that. That's a good point.
Thank you.
Pasquale, Charletta, or next.
Hi, Helen. Nice to see you. Thanks for the talk. It was a long time I haven't seen you.
Hi.
I wanted to ask you just a few quick questions, which is, one is like, do you have any development in mind where you want to couple aggregation and diffusion, or like coupling some kind of transport equation with smoluchowski or other kind of coagulation equation to also get into the formation of the plaques? And second is that if you have thought about other diffusion equations, like I don't know, an Allen-Kahn or something other potential to explain the transport dynamics, or if there is any significant difference between this and fisher-kolmogorov. You think that it's the basic ingredient is already there? Thank you.
No, this is a great question. It's great to see you. So we did solve the smoluchowski model on the network. So that's possible and relatively cheap. So you have 83 nodes and you have a length of, so I think the student who did that looked at length of about 200. And I have also run it on my laptop. So that's doable with a length of smoluchowski length of 200 times 80 nodes. That's not a whole lot. That's less than 2000 degrees of freedom. If you want to run it on a finite element mesh, we haven't, because that's going to get more expensive. So then you can actually look at smoluchowski on the brain mesh quite nicely. We haven't looked at other diffusion equations, and I think that's probably a good point. So the one that we use here is just the simplest, right? And I'm sure you can make that more complex, especially as more data become available. It's probably worth going back a step and refining. I think it's a great point. And especially with your community, coming from a math background, you probably have a much better understanding which math equations would describe phenomena that the simple models can't capture at this point. That's a good point.
Thank you.
Davide Riccobelli. You're next.
good morning. Thank you very much for your presentation. And I have some curiosities about the mechanics of the brain, in particular on how these misfolded proteins affect the mechanics. So do you have any idea on which is the effect of these proteins on the mechanics of the tissue and also of the cells, if they affect in somehow how the single cells behave?
So this is a great point, and I didn't touch on that because the model we looked at here is very macroscopic. So you're asking for the mechanism by which the misfolded proteins breaks down the brain, and you see this atrophy, right? And so if you look at the cell, so you have an axon as the brain, the cable in the brain. The axon is made of microtubules. So these are the stabilizing long proteins that actually make up the length and make it really stable. So an axon can be up to 1 meter long, going from the brain all the way down your spine. So it can be very, very long. And the reason why it's stable is because it has these microtubules, and these microtubules are cross-connected, and they're cross-connected with tau. And that's exactly the tau that we're looking at. So if this tau clumps up, the tau can't cross-connect the microtubules. And so then they get free, and then they actually disassemble. So if you look at the protein kinetics of microtubules, they become unstable without tau, they break down, and then the axons, the main cells of the brain, would break down without that stability. So that's what explains the connection between what happens on the tau level and actually the atrophy, the mechanics. And so you can build that into the model. We've looked at axons, at very small single scale axons themselves, and modeled how they break down. But that's a huge step from just the cell to the whole brain, right? So we haven't made this very connection. We've looked at it at a small scale, at the larger scale, but now you would also need to connect, and that's a good point. Yeah.
Oh, thank you very much. For me, it is very interesting because I'm also working on a similar problem. Have you ever, do you have some papers that are published on this topic?
Yeah, so I would suggest, can you find my email address somewhere in the internet, and then you can email me. We can follow up one-on-one, and I can.
Thank you very much.
Yeah, great. Thanks. Great question.
Luca, Fede?
Can you hear me?
Yes.
Yes.
Okay. Professor, thank you very much for the very nice talk and full of insights. So my question is related to the mechanical problem and the boundary conditions that you use for the mechanical problem, especially in relation with the fact that you have a shrinking domain, and how do you account for this, and how is this space filled by other fluids or other elements during the process of atrophy? Thank you.
That's a great question. And actually, I haven't talked about this much because it is actually a real problem. So if you look at these simulations that we've done, the 2-dimension simulations is easy to kind of fix because we just fix the brain stem. So we fix this in the middle, and then you can see it shrinking towards that point. And that's why this simulation on the coronal slice works very well. If you do it on this slice, it's very difficult to fix in space, and there's a lot of rotation if you do. And actually, it's not really fixed in space. So that's what's creating these artificial rotations if you shrink, right? And if you look at these simulations, we were struggling a lot with these boundary conditions, especially in 3 dimensions. It becomes very tricky because if you just shrink, it doesn't just pull itself together because you need to fix some points. So we're trying a lot of points, and obviously wherever you fix something, you introduce a singularity into the equation. And so you have high stress concentrations. So what we've done here is we've just fixed something very far away, and then we've cut that off because we believe that's probably not what is physical. But that's a real important problem. And I think it's a problem always in boundary conditions. When people work with the heart, you have the same question, where do you fix things in space, right? You always induce some artificial singularity. You need boundary conditions to solve the problem. So in this case, it was just simple because we used the symmetric problem to fix the brainstem. But we had to play a lot with that. And actually, yeah, that's a good question. And it shows that you actually have probably tried the same. So if you want to talk some more, I'm happy to kind of meet or talk via email and follow up on that.
But is this space going to be filled by fluid, or is just then stay?
Yeah, it's not empty. So it's just filled. I think someone just asked about the fluid. There's a lot of fluid in the brain, which initially wasn't clear to me. There's also a lot of circulation of fluid in the brain. So it's not static. And so there is this outer space is actually fluid, which is nice because if you hit your head, it's always damped with the fluid, right? It's a good thing. But you'll have a lot more fluid when there's a smaller brain, obviously.
So then, as Paolo Zeni was saying at the beginning, then coupling with the fluid would be perhaps a better way to account for the
and also makes the mechanics very and it makes the mechanics very difficult to measure because you have this huge increase of fluid. If you were to really measure the mechanical properties, you'd also have to account for all of a sudden the balance of the amount of fluid and brain in the brain when you really measure the stiffness, right? So it's kind of tricky.
Okay, I see another question. Giulio Bonifazzi.
Hi, Ellen. A quick question. So part of the issue of the curves of the sigmoid shape that you show, they don't consider the potential interaction between different species, right, in the model. So how would you account for them? Are you planning to account for them? Or, I mean, this is something quite major, right? As well as the accumulation of misfolded proteins and the formation of oligomers is an accruing process over time. So you don't have a smooth transition by stages. It's much more chaotic. So how this potential correction of your model might actually influence the outcome of your current predictions?
This is a great point. So I'm working with Alain Gorelli. Many of you know Alain. So he actually has a model that couples both tau and amyloid beta. So he looks at really both building up, and then he looks at what the interplay between the different species is and how that actually accelerates and connects these different curves. So if you're interested in just this very coupling, Alain has looked at in just a recent publication, and I think that's also a very interesting question to ask. It's difficult to tease out the data, as you probably know from just brains. A lot of people study in a dish, and the data in the dish doesn't necessarily translate into real brain. So I think the fact that we have these models is great. We can play a lot of what if. So Alain has done a lot of what if this increases, how would that affect the coupling? How would that accelerate the process? I think now it would help to really have more good data to actually see to which extent this coupling, like exactly what you're saying, is relevant, because I agree, this is very simple models and very early stage in that sense. Good point, yeah.
I don't see any other hands raised. So let me thank Ellen for this very great and inspiring talk. Of course, there are plenty of open issues, but yet what you've been able to prove already is something really impressive and extremely important. I'm sure that this subject will attract the interest from many, many researchers because of its complexity and because of its interest, social interest. So Ellen, thank you very much again for your talk, and all the best.
Yeah, thank you. Thanks
Goodbye.
for organizing it, inviting me. Thanks for the great questions. Very stimulating. Thank you.
Great honor to have you here.
Thanks.
Goodbye.
Bye.